Now we will discuss the chapter 4, the three-dimensional structure of proteins. Okay, so many conformations are possible for protein. This is due to the flexibility of amino acid linked by the peptide bond. So you have the different possibility for the peptide bond to form the that means different amino acids can connect to each other. Okay, so for the DNA sequence, you have ATCG to synthesize the nucleic acid chain. For the peptide bond, then each position you can have 20 possible amino acid to add it to the next one. So that's why you have many conformation. All right. And then each peptide bond, they can also form different conformation. That's because the location of each amino acid and also their side chain can also provide the interaction with each other, and therefore even the same sequence of amino acid, they could also have a different conformation. But for all these different conformation, right, only at least one major conformation that has a biological activity, and that would be the native conformation for that specific protein. Okay, that's called native conformation. And then the level of protein structure, that will be the primary structure, secondary structure, tertiary structure, and these quaternary structure. So for the primary structure, basically now be the amino acid sequence, all right? The amino acid sequence, and the primary structure can be determined directly by the DNA sequence, all right? So the DNA sequence that you see that can be translated directly to the amino acid, and then that would be the primary structure that you can translate directly to the primary structure. Okay. And then again, the N-terminal always begin from N-terminal, and then to the end will be C-terminal. And the primary structure sequence of protein can determine the three-dimensional conformation. And change just one amino acid, okay, that will alter the biological function. And the most important example that will be the hemoglobin, all right, associated with the sickle cell anemia, because they change the number 6 amino acid in the peptide, and then they change the biological function. Okay, so that's the primary structure. And the secondary structure. So the secondary structure basically is a hydrogen bonded arrangement of the backbone of a protein. So this hydrogen bond basically come from those peptide bond, all right? So that'll be the bond, for example, between the alpha carbon and amino nitrogen in residue, and the bond between the alpha carbon and the carboxy carbon the residue. Okay, so that will give you this that contribute to the secondary structure. Okay, and the two are commonly seen secondary structure. One is alpha helix, and the other one is a beta sheet. All right, so the alpha helix, okay, the alpha helix here. So you have the CO of each peptide bond is hydrogen bond to the NH of the fourth amino acid. All right, so basically the hydrogen bond between those peptide bonds contribute to the secondary structure. And in this case, so that will be as you can see, the CO bond here. All right, they will form the hydrogen bond with every fourth, the hydrogen bond with this hydrogen of the NH here. All right, and then the CO number 4 NH, and CO will be number 4 NH. All right, they form this hydrogen bond. Okay, so that gives you this. This is the hydrogen bond within the alpha helix. Okay, It's an alpha helix. You can see those hydrogen bond between the CO and NH. So the hydrogen bond, all right, come from the peptide bond, all right, to stabilize the secondary structure. Very important to remember this. So the primary structure, so we have the peptide bond to connect to each other to give you the primary structure. And then the secondary structure, so you need the hydrogen bond between those peptide bond, okay? So alpha helix, okay? And several factors can disrupt an alpha helix, okay? So for example, proline create a band because the restricted rotation due to its cyclic structure, and two, its alpha amino group has no NH for the hydrogen bonding. So that's a proline. If you have proline, that can disrupt the alpha helix. Okay, remember the proline because they don't have their primary amine. They form this ring, the secondary amine. All right, so that's why that will disrupt the alpha helix. And the strong electrostatic repulsion caused by the proximity of several side chains of the light charge. So for example, positive charge or negative charge, lysine arginine positive charge, and this negative charge, and then they can produce the strong electrostatic repulsion, and therefore that can disrupt the alpha helix. And also the stereo crowding caused by the proximity of bulky side chain like valine, isoleucine, threonine, and that will also disrupt the alpha helix. Okay, and the second type of a commonly seen secondary structure that will be a beta pleated sheet. Right, so the polypeptide chain lie adjacent to one another. It could be parallel or anti-parallel. And then the R group alternate first above and then below the plane. And each peptide bond is planar. And CO and H group of each peptide bond are perpendicular to axis of the sheet. And then you form the hydrogen bond between the adjacent sheet, all right? So just like this. So you have this parallel, anti-parallel. So this is from N terminal to C terminal and N terminal to C terminal. And then this hydrogen bond in between between these the two different sheets here, all right? So that's the hydrogen bond between these two different sheets. For alpha helix, okay, area CO will form the hydrogen bond with the force NH. And but for the beta pleated sheet, so basically you have this hydrogen bond between these different sheets here, all right? So it can be parallel, anti-parallel. Okay, this is the beta pleated sheet, the three-dimensional form of the anti-parallel beta pleated sheet arrangement. Okay, this is anti-parallel. And so this is alpha helix. So you can see this peptide, the CO from the peptide bond form this hydrogen bond with the NH, all right, with every 4 amino acid. And this is the hydrogen bond between these two different sheets, all right, and then the CO, NH, NHCO, okay, to form a hydrogen bond. And they have fibrous protein, right? So fibrous protein basically are insoluble in the water and the diluted soda solution, and they play the important structural role in nature. So they have the polypeptide chain organized approximately parallel along a single axis. All right, so be similar like this. All right, like a collagen here. Okay, so that's fibrous protein. All right, example be collagen. Okay, and globular protein that can which are folded to a more or less spherical shape and tend to be soluble in water and soda solution. and the polar side chain are on outside, all right? And the non-polar hydrophobic side chain will be buried inside, okay? And nearly all have a substantial section of the alpha helix and beta sheet, okay? That's globular protein. So this is the fibrous protein and globular protein, right? And the tertiary structure of a protein is a three-dimensional arrangement of all atoms in the molecule. In the fibrous protein, the backbone of protein does not fall back to itself. It's an important aspect of tertiary structure not specified by the secondary structure. And the interaction between the side chain play important role, okay? So like I just mentioned in the chapter 3, so the hydrogen bonding now between the polar side chain, hydrophobic interaction, all right, between the non-polar side chain, and then electrostatic attraction between the side chain opposite charge that give you ionic bond, all right? And then you have the repulsion, that's of the like charge, and plus the disulfur bond, all right? That's a covalent interaction, disulfur bond between the cysteine. So these interactions contribute to the tertiary structure. So you have non-covalent and covalent. Covalent would be the disulfur bond, and non-covalent, you have the hydrogen bonding between the polar side chain, hydrophobic interaction like a van der Waals interaction that's between the non-polar side chain, and then you have this electrostatic attraction, the ionic bonding between the opposite charged amino acid, and also the repulsion, that's a like charge. So this can show you that the interaction, the disulfur bond between the cysteine, all right, here, and then you have a hydrogen bond, all right, and then electrostatic attraction ionic bond, okay, hydrogen bond, and then you have hydrophobic interaction, okay. So these interaction contribute to the tertiary structure. And so the tertiary structure is the arrangement in space of L atom in the polypeptide chain, and the quaternary structure, basically the association of polypeptide chain into the aggregation, okay? So then the two large class, that's fibrous protein and globular protein. So that's basically the structure of the protein from the primary, secondary, tertiary to the quaternary. And the primary basically is linked by the peptide chain, peptide bond, and the secondary stabilized by the hydrogen bond between those the peptide bond, and tertiary structure stabilized by the those side chain interaction. So you have the hydrophobic interaction and the hydrogen bond between the polar side chain, and the ionic bond between the opposite charged amino acid, and also the disulfur bond between the cysteine. Okay, so that contribute the tertiary structure, and then quaternary structure, that would be the many subunits, many peptide. They form the interaction, and then to produce to give you this quaternary structure. So it could be dimer, trimer, tetramer, all right? So that's the quaternary structure. And then for the protein structure, the three-dimensional structure, there are different ways to determine the protein structure. So you can use the X-ray crystallography. So basically, you have to use you have to crystallize the protein, and to do this, then you have to use the genetic engineering technology to clone the specific gene, right, into the bacteria, and then you try to express the protein, and then you crystallize the protein, After you crystallize protein, then you will be able to expose this protein to the X-ray to obtain this diffraction pattern, and then you'll be able to interpret the protein structure. Okay, so like this picture here, that's X-ray diffraction photograph, okay, of the protein, so that you be able to interpret the protein structure. And then also you can use the MMR, that's two-dimensional MMR, and then to determine the structure. So these are some technique people use in the field to determine the protein structure. And this is to show the myoglobin, which is a single polypeptide chain of 153 amino acid, right? And then you have a single hemic group in the hydrophobic pocket here, hemic group here, right? And then you have eight region of alpha helix and no beta sheet. And this is the structure of myoglobin. And then the oxygen bind to that hemic group of the myoglobin. And denaturation and the refolding. So non-covalent interaction, okay, they are weak and they can be disrupted, okay, non-covalent interaction. So non-covalent interaction, so like we just mentioned, so you have the hydrophobic interaction, ionic interaction, and the hydrogen bond between the polar side chain. Those are the non-covalent interaction. So they are weak interaction, can be disruptive. And when we say the denaturation, that means it lose the tertiary structure, okay, unfold the protein. And then to denature the protein, so you can use the heat, right? Use the heat, or you change the pH. These physical change, okay, can these change of the environment can disrupt those non-covalent interactions, okay. And also you can use a detergent like SDS that can disrupt the hydrophobic interactions between those non-polar amino acids. And then guanidine, urea that can disrupt the hydrogen bonding. So the hydrogen bonding that's between those polar side chain, all right? Those polar amino acid, those polar side chain, you can use the urea and guanidine to disrupt their interaction. And then the reducing agent, okay, like mercaptoethanol or the DTT, those are reducing agent because they can reduce the disulfur bond between the two systems. Okay, so these are commonly used technique to denature a protein, and also the technique that you use to treat the protein before you perform the gel electrophoresis. Okay, the SDS page gel electrophoresis to determine the protein molecular weight or to see the protein quantity. So you have to treat the protein sample with these different methods so that you'll be able to run the protein on the SDS page gel. And this is just to show you the native conformation, which is a functional conformation, and denatured protein. So denatured protein, heat, pH, detergent, like SDS, urea, guanidine, and also the reducing agent like mercaptoethanol and the DTT, so that you can break all the interaction and give you the denatured protein. So the coordinate structure, the association of the polypeptide monomers into the multi-subunit protein. So you have dimer, trimers, tetramer. Okay, these are the coordinate structure. So they have non-covalent interactions, electrostatic, hydrogen bonding, hydrophobics. Okay, those are from the tertiary structure. as well. So the structural change occurred during the binding of small molecules. So basically, that's called allosteria behavior. So when you bind something, the protein, when you bind something, then they will change their structure. Okay. So protein folding. So to predict the protein structure, yes, you can predict it, but it's difficult, right? But here is just a reference, the link. If you are interested, you can go to this website and then give the amino acid sequence. Then they can predict the protein structure. Okay. So basically that would be the chapter 4, and then the correct folding. Yes, because that, yeah, just a second out there. So the basic protein, as soon as it is translated, all right, when you synthesize the newly synthesized protein, as soon as they are synthesized, you have to go through the uh, the chaperoning, and then this protein will help to fold the protein into the correct conformation, and they will become functional. Okay. So that's why it's important that you have to fold the protein correctly. If you don't fold correctly, then you could form the aggregate like this. Okay. And then you need the sharpening to sharpen it to fold the protein into a correct conformation and then be functional. Okay, so that'll be the end of chapter 4. All right.